Hi there to everyone who clicked on the video. The video I want to talk about my recent research, which needs a bit refinement, but anyway we can talk about it. Here in the video is about the character evolution of the genus Burmanniaceae in family Burmanniaceae. And before we go any further, I want to let you know what is the Burmanniaceae is in this particular family, Burmanniaceae. What interesting about this family is that it's a mycoheterophytic plant family. All the 13 genera listed here is all mycoheterophytic plant that can do the photosynthesis and rely on the fungi associated with the fungi to bring the nutrient to them. But in the Burmanniaceae, there are some of them that still be able to do the photosynthesis, which is kind of weird to like only one genera here, only one genus here among 13 of them, and is distributed across the globe here, especially in the tropical area, like in the forests. There are about 68 species, except the species around the world. And the objective of this research is to observe, to investigate the morphological change within the process of evolution of this Burmanniaceae, and to describe why is happen like that in the evolution. And here, just a brief morphology of the Burmanniaceae, so you guys can follow me later on and not get confused. Oh, sorry. And oops, get in the way. Here is the Burmanniaceae plant. The whole Burmanniaceae plant consists of only a flower, an inflorescence, and a stalk of the flower. Here a little leaves here, but in some species there are like robust leaf, but this one is quite small. And when you zoom in the flower, here you can see this fluffy thing is wings, flower wings of the Burmanniaceae. And when you look at the top part, the white parts here are the tepals, and inside here, when you zoom in, is in this floral tube. There are 3 stamens. Each one have these appendages. On the top is called apical crypts, and at the bottom is a basal hanging spur. And in this research we conducted about 28 plot 5 from the Burmanniaceae and the Burmanniaceae family to do the phylogenetic reconstruction. And how we study the evolution, we start with the plant material. In the picture here, 9 of them is collected in Thailand as a fresh specimen and keep it in the fridge. Some are in the silica sand and gels, but the rest are from the Gen Bank and provided by the National Biodiversity Center also. So for the fresh and dry specimen, we extract the DNA using the kit. Then we amplify the DNA using these marker ITAs 18S or DNA, and this NAT1BC, which is the mitochondrial, and the RBCL is the plastid, or in this is the colopod, or the reduced one, which is for the fully microscopic plant that don't do the photosynthesis, but there's still the positive there. And then we send to the company to do the sequencing for us. Then they give us the result of the sequences. Then we have to do the alignment by ourselves using the MAF, which is the online platform. And then we do the phylogenetic reconstruction, or do the phylogeny. Here we're using two methods, is the RAXML to do the maximum likelihood method and the Bayesian inference. We use Mr. Bayes on Cypress, which is the online platform also, because it's taking a lot of computational results to do, so it's quicker to do online. And then, because I also do the traditional taxonomy about this genera, so I have the information about the characters of the morphological characters of this plant. Then we have to code it into the metric so we can feed it into the program, which is the R. Here I mainly use these two to do the work, these packages, mainly the phi2. Some of them are just to visualize, and the if to read the map, and then to read the tree. And for the reconstruction of the character state, we use this stochastic mapping method from the phi2s also. And here the result for the phylogeny. First one start with the 18 or 18S RDNA, which is from the nucleus. Here it is origin of the Burmanniaceae, 28 of them. The 18S get quite moderate in terms of the support. Some are high, some are low, but I would say is pretty decent for just early one region. And the next one is a little better in terms of the support and different in terms of the topology. Here is from the ITS region. The support is, some of them are greater, and the protein is different. So it's quite a nice region to be added. The next one, this one is from the mitochondrial NAD1 BC region. The support are quite low compared to those two previously, and the topology is similar to all those, the ITS. And the last one, the RBCLA is a part of RBCL gene, which is quite a big gene, but only one part is the constant part, so the resolution here is quite low. And you can see it's from these are packed up here, and this long bench is from, because this indicate how different each taxa are. So the Burmanniaceae now is transferred to the Campylosiphon, not in the Burmannia anymore. So it have quite a really distinct, so it's showing long bands here, but all the Burmannia are packed up here. So this is not useful, so we have to discard this information for the further analysis. And here, just to show the combined tree compared to the ITS. So the combined tree is influenced more by the ITS region, so it's similar to the topology of the ITF region. And here, the Bayesian and the maximum likelihood result is give us the like almost identical in terms of topology and support, because all the support at quite low in the maximum likelihood is grouped here in the polytomy part of the Bayesian analysis. So it's quite concurrent in two methods for the phylogenetic reconstruction. The next part of the result is ancestral state reconstruction. I start with the stem color. So here at the node, it shows the probability that the ancestor of the Burmanniaceae tend to likely be the green for the stem color. Then it evolved into the purple here for the ancestor of Burmanniaceae and Itoena. Is in the same class as the leafy one. And here is purple in the white one. Here is ancestor of the cryptopetalar clade in the Nepenthes here, and other white independently evolved. So it's from green to purple and from green to white, not from the purple to white, or not in the other way. And here, the size of the basal leaf. It starts from lacking here, like in the tree is showing, and then develop into more robust in some clade, and quite a fill for the smaller rosette leaves. But at the ancestor of the Burmanniaceae is show that it should be lacking, like their sister genera. For the flower color, is ambiguous here, so we cannot tell apart which one should be the ancestor. But in this certain clade, it shows quite a clear evidence of evolving from purple into white, which is in the same way as the pearl ant or the table color, like in the white one, is tend to have the yellow one evolve into. And here in petal present, some of them, like in the Burmanniaceae oblonga, is absent. So in Burmanniaceae, the ancestors show that from the start, ancestral state of the Burmanniaceae is from the present to absent in some of them independently. The next one, sorry. The apical crates present, similar to the inner perianth, is from the present into the absent, scatterly, not from the single origin. And the basal spur, we cannot tell from here because it's 50-50% and is all scatterly evolved. And this is the interesting part also, the flowering spans. So at the side, but I separate into 4 discrete characters here from the board: medium, narrow, and lacking. And at the origins of the Burmanniaceae, we cannot tell because it is ambiguous here at the node at the origin ancestral state of the Burmanniaceae. But here, in the same case at the white flower, it evolved into the bigger wingspan. So later I plot it into the continuous characters, not a decrease. So for the Burmanniaceae, You can see the darker color here is indicate that the Burmanniaceae tend to evolve into bigger wings, especially in this clade. Not like at the ancestor of like further down the Burmanniaceae in Burmanniaceae, which is already they didn't have wings. And here the summary. So I plot the icon into basically the same tree, but everything sum up here. And for the discussion, so it's quite amazing to see here that we tend to think that the evolution tend to be only in one way, like from the photosynthesis, for like able to do the photosynthesis to not able to, but in Burmanniaceae is keeping that characters form the further down ancestor in Burmanniaceae, and some of them still keep and evolve in back into relying on more on the photosynthesis. I think this is my theory that maybe the ancestor of the Burmanniaceae is living in more shady area, like in the dense forests understory. But at the border of like the forest, which is some like grassland here, that plant evolve, that Burmanniaceae evolve into having more robots in the open area, which is quite, interesting for the plant to evolve back into what it used to be. And for the color of the flower, I guess it's from the perspective of the pollination syndrome, because like show in the tree, the white flower tend to have a bigger wings, which means it can like spot easier in the dark because it likes the color, and the target for the insects or whatever is bigger. Or maybe because the wing also persistent when the flower becomes a fruit. Maybe is help the disposal of the seed, like is kind of flapping when the wind blow and the seed is falling all over. And like in the more purple color flower Burmanniaceae, they tend to be in the grassland, which is green. The purple, the vivid purple is contrast to the green grasses. So it's easier for the pollinator like bees. And the different color between the table, which is the top part that the insect can land and do the work, like pollinate and maybe sipping some nectar, or different colors from the race of the flower. And here the conclusion, the base T are the combined T in terms of the support, and the topology is similar to of the ITS, but in here we use the ITS because there are more text available here than the combined one. And the morphological characters of the Burmanniaceae tend to independently evolve, but in some certain characters like the wings, the flower, the perianth color, the size of the wings evolve into a certain trend, like is co-evolving into particular appearance. And thanks to everyone here sharing this picture. For any support, and if you want to save this plant, please save the West Grassland. Thanks for accompanying my video. Bye.